Hi, this is Sahil from Radianz Academy. So, in the last presentation, we learned something about artificial reproduction in plants and its various types. So, in this presentation, we will learn something important about micropropagation, cultural techniques, its advantages and limitations. So, let's come to the first topic of this presentation. What is micropropagation? Micropropagation is the practice of rapidly multiplying stock plant material. So, there are some interesting facts about this process of micropropagation which will amuse you. Some of them are no cuttings from the plants are taken. Instead, few cells or tissues are extracted and then they are cultured in a suitable nutrient environment to produce new plants. This process of micropropagation helps to produce many progeny plants by modern culture methods. In this sentence, the word progeny is very important. Progeny means offspring or new generation plants. You can notice in this figure how new generation or progeny plants are being produced. This process of micropropagation also helps to multiply genetically modified plants. As you can see how a rabbit has been converted into a cat, how a man who is running has been converted into a superhero, and how a child scoring bad marks in the exam has been converted to a happy child who is scoring good marks in the exam. These three are all examples of genetic modification. Now we'll come to the next topic, that is cultural techniques. So, does anyone know what are the cultural techniques? Plant tissue culture involves micropropagation, which we learned just now, and plant cell culture. Plants usually reproduce by producing seeds. This process of tissue culture produces a variety of plants which are similar but not identical. Now we'll come to some advantages and limitations of this process of cultural techniques. So, what are the advantages? This process of cultural techniques helps to quickly produce mature plants. In this process, there is no needs of seeds for reproduction, only a small amount of plant tissue. Can you even think of chicken without eggs? No. The most interesting and the most important point in these advantages is that only a small amount of initial plant tissue is required and not the whole plant to produce new progeny or offspring plants. Now we'll come to some limitations of this type of cultural technique. So, first limitation is that it requires a lot of scientific expertise or a scientist team to produce many offspring plants. Next point is here of limitations is that it can cost more money. This is a very important point. That is, before being cultured or before being reproduced, the material is screened or checked. Failure to pick up any abnormalities could lead to new plants being infected. These are the two pictures which can show how the plants have been infected because the material was not screened. Last point of limitations is that it's not easily applicable to the remote areas or where the network facility is not there. So I hope you like this video and please don't forget to subscribe. Bye.